Hi, my name is Caleb Ellington. I'm a senior studying bioengineering and computer science at UW, and I work in the Baker Lab on structural refinement. And today I'll be speaking about my work on deep learning applied to unreliable contact prediction in de novo or computer-generated protein models. So protein folding is a very hard task, and we do it for a lot of reasons. If we could instantly know every protein structure, we would have therapeutics that don't elicit an immune response, new types of foods, endless new therapies and industrial processes, and of course, a COVID-19 vaccine. Unfortunately, we aren't even close to that point right now. The current standard for protein folding is Rosetta, which is pretty good at making small single domain protein folds, but that's where the practice of computationally refining a protein structure becomes very important. AlphaFold, RaptorX, and TR Rosetta are technologies that make tweaks to a protein structure until it looks closer to how it would in real life. Unfortunately, it doesn't always work perfectly, as you can see from the bottom region of this protein right here, which is much more chaotic than the rest of the structure. Luckily, a new tool called Deep Accuracy Network, or DeepAccNet, developed by my mentor, now Hiranuma, can help predict the error of a given protein structure. Um, so another way of visualizing a protein structure is with distance maps, where instead of looking at proteins as a big point cloud, we can visualize them as the distance between all of the points. Here you can see a predicted distance map and a true distance map for a protein structure used to calculate the error, or the histogram, of the prediction in terms of how the distances need to change to be closer to the true structure. Now's deep accuracy network predicts the error when handed a modeled structure, which can then be used to calculate the unreliable regions of a protein, which are most difficult to model. My research specifically focuses on improving the structure within these unreliable regions by predicting contacts or short distances between individual amino acids, which is very useful information for other structural refinement tools. The way I've done this is by taking all available information about a protein, including the histogram and unreliable local region data, and feeding it into a very deep residual network. By training the network with known protein structures, it can spit out a distribution of probabilities that tell us how likely the distance between two amino acids falls into a certain category, whether that's 0 to 5 units, 5 to 6 units, and so on. I then take the maximum probability bin and assign it that bin's distance value. In many cases, the results look promising. These are the same distance maps as before, but showing only contacts under a certain distance threshold. For protein TR829, my network is able to pick up on certain structural motifs that the standard structural refinement protocol cannot recognize in its first through 50th iteration. Additionally, when comparing the accuracy of amino acids that are near neighbors in sequence, my network outperforms the contact prediction on almost all of 74 refined protein models that the network has never seen before. I hope to continue improving the precision of this technology and apply it to the current structural refinement pipeline to edge closer to a world with accurate protein structure predictions. I'd like to extend a special thanks to Now Hiranuma, as well as the whole structural refinement team, for opening my eyes to this field and making the IPD such a wonderful place to work at. I'd also like to thank Lukey Goldschmidt and David Baker, who have been integral parts of my development as a researcher. Thank you.